I'm sorry we all couldn't be together in person today to meet, because I feel like when giving a talk, it's also always helpful to have feedback. So please do ask questions in Slack, and if we don't have time, I'm happy to continue discussions afterwards. Okay, so today I want to give you kind of an overview of different applications of computational topology and how useful it can be to different areas of primarily molecular and cell biology. But of course, there's a very large range of possible applications. So yesterday, actually, there was a paper or an article that I pulled. It was on Twitter. So you can see that the publication date was July 10th, 2022. And I think that we all agree that mathematics, in particular geometry and topology with machine learning, really enhances what we can do for applications and in particular for biology. And so if you haven't seen this, this was actually written by a previous professor who was at Imperial College, so within the London research ecosystem. And in this article, they talk about many different biological applications ranging from viruses to climate. So mathematics really, as they say, at its core, is the science of patterns, and computational geometry and topology are very useful mathematical areas. I hope to convince you today of studying such patterns. So as I said, my talk is going to first introduce some very kind of coarse ideas from computational topology, and then I'm basically going to give you a very quick, I think I have something like 8 possible applications. So it's very quick. There's going to be references throughout, so this is more just a taster for how topology can be useful in biology. So I want to convince you that data has shape and shape matters. We're all, maybe I don't need to convince you, that's why you're here. And this is, if you are just starting out as a student, I think review articles are probably the best way to get, in addition to talks, right, are the best way to kind of get an introduction to a field. And so with Chris Bick, Elizabeth Gross, and Michael Schaub, we wrote this article that will appear in Science Review, and it discusses different data structures within computational geometry and topology that can be useful. So basically extending from a graph, vertices and nodes, vertices and edges, to higher order networks. And you can think about this in terms of dynamics and structure, as well as statistics. So some of this that we discussed in this review, it has some kind of statistical networks and tensors. So if you're interested, have a look. It's also available on the archive. Today's talk, I'm going to basically advertise the next talk, which is, please go to Catherine Benjamin's talk, which is an introduction to one parameter, persistent homology. And I think computational topology, one of the strengths of it is it's a multi-scale summary of data, and we can extract via topological invariance features of this data. So probably the most developed and prominent tool in the field is persistent homology. And one of the strengths of it is that we have stability theorem. So it's robust to perturbations of the input data, which means that the output just perturbs a little bit. And one of many reasons why you may be interested at a machine learning conference or school is that it's compatible with machine learning statistical techniques. So if I give you if I ask what's the topology of this point cloud, and I give you these points, it's a subset of R2, and I say let's use let's use the Euclidean distance, then the first thing we would do is build this higher order graph called a simplicial complex. And one way, there are many different ways to build a simplicial complex, and that depends on this metric, right? And so with the Euclidean metric, what we can look at is effectively building, looking at where at some distance these points intersect, or a pairwise distance. So we can look at where different points, data points are intersecting, and look, so you're building a nerve of this cover, and you can then compute what's called homology. So this is a topological invariant where you can measure different notions of holes. So zero-dimensional, so H0 is connected components, and H1 gives you one-dimensional holes, so loops. So you can kind of already see what we're gonna, we pick some distance epsilon here, and we can see that we went from 20 data points to, I think 10, 1, 2, 3, 4, 5, 6, 7, 8, 9. And so then at some epsilon, you continue, and as you continue to increase this epsilon parameter, we can see that many of these connected components, these clusters, are merging. And then ultimately, then we see there's a small one-dimensional loop here. And then finally, by squinting at the data, we can see that there's a one-dimensional hole, there's a loop. And that is, that persists over a long parameter range. So this, and then finally, when epsilon gets so large, then this hole completely gets filled in, and we only have one connected component, which is an infinite feature. So this was persistent homology in a slide, and this is kind of the summary of it. Again, Kate will properly introduce this and all the building blocks in the next talk. So maybe to mention that Carlson, Greist, and many others have been working in the field of topological data analysis, and they started this. That's kind of how I was introduced to this, and it was before we could do computations. Right, so you start with some data, and you build a filtered simplicial complex, approximating the shape of that data. We compute these holes, computing homology, and what we get out is a multi-set of intervals called a barcode. Now, 2017 now feels very long ago, and this was right when there weren't as many fast implemented algorithms. So we kind of went through the various different software. Again, I think Kate's going to mention what is the state of the art in the next talk. Those of you that are eager, probably the one that is the fastest is RIPSR, and I often use them. If you look at Henry Adams or Peter Bubinick's website, you can look at, and they have a very long list of various software. So once we have a barcode, we'd like to be able to interpret what these topological features from the data. And so one way you can do this is through vectorization. Again, Kate is going to go through and give a much thorough definition of all of this, but the general idea is that you build some explicit feature map on your barcode, your persistence barcode or persistence diagram. These are equivalent. One is just intervals where you plot them as a multi-set of bars, and the other is where you have an X, Y, so then you have points in the X, Y plane. But it's the same, it's an multi-set of intervals. Then you can take this persistence diagram, this multi-set of points, and project it into a vector of Rn, and then build some suitable Hilbert structure on that. And so two examples are persistence images, and this is where you build a Gaussian on each of these. Well, again, Kate's going to explain it, but it's a way to vectorize. Both these are ways to vectorize, where you can then perform statistics on this. So as a kind of pictorial example here is these are each of the bars in your barcode, and you build these tent functions and then collapse it down, and you have the most persistent, the second most persistent feature. So these are second most persistent bars. And then you can take averages. So you get an average persistence landscape, which we use later in applications that I present. There's also kernel-based methods. So this is where you're, I mean, I don't need to explain. So there's many different examples. And then I guess the thing to highlight really, which has changed since I got into this field, is that we can compute very fast. And that's just wonderful to have so many useful and efficient algorithms for one parameter persistent homology. So that is my introduction to persistent homology. And now I want to motivate the problems that we're looking at. And the first thing is that if we look at, you'll see this image again. So this is a cell, and we're thinking in molecular biology. So if we think of a cell a cell has different compartments and it has many different molecular species, such as proteins. And a cell has to sense what's happening on the outside of the environment and relay this information into the brain, to the nucleus of the cell, and then turn genes on or off, and that governs cell fate and cell decisions. And it's really remarkable because all of our cells, each second we've had millions of cells being produced while you're listening to this, and we all have the same DNA in our cells. Yet we have many different cell types and different molecules, and proteins are expressed and genes are expressed. The function of cells are very different. So I can give you an example, like if we think of a blood cell, this is very different than a neuron. And the location matters a lot. So for each of these dots here is a cell in a cancer. And what they've done is it's the same type of cancer, but they've changed the condition, the environment, and it's affected where cells are located just by changing, whether it's via a drug or some other type of condition. It can affect the spatial patterns. Remember, the first kind of motivation is that we need maths in biology because biology very much is patterns. So is machine learning. So these patterns matter a lot for these markers of different cell types. And this is in particular, we're thinking of immune cells. So the cells are really kind of the key building blocks in much of biological processes. And so this middle figure, that the panel here, what I want you to take away is that it's very complicated. We have many different molecules that are interacting. These look like they're assigned some color, and that color is then describes what they call a signaling pathway. And the molecular, these protein interactions can be modeled mathematically. And when we look at different mathematical models of this, there's various ways that we can analyze it. So I want to the rest of my talk now is different examples of how topology can be useful in biology. And I'm going to start first at thinking of these molecular interactions, these proteins, and we can model these molecular interactions as biochemical reactions. Okay. So it's the same motivation. We pick one signaling pathway, and we can write out with some assumptions that some of these molecules, these proteins interact, and a certain number of them interact, and they interact at some rate, kappa I, and then they form, this is the reactants, and this is the products. And and they form a system of chemical reaction networks. Excuse me. And so this is meant to be a model that is a hypothesis. It encodes a hypothesis of what is happening in our molecular system. And we're going to work in the deterministic setting for the first example, which assumes mass action kinetics. And that is where the rate of the reaction that the reaction proceeds is proportional to the product of the concentration of the species. So, so we have these rate constants. We have our species, the xj, there's n of them. And we have these stoichiometric coefficients. These are natural numbers. And we have a certain number of species that interact, then that from the reactant to products. And with mass action dynamics, we have a formula which we learned in the first or second lecture of mathematical biology, that we can look at the concentration. So instead of thinking of these species in capital X, we can think of the concentration of these species and how they change over time. And these can be modeled as a polynomial dynamical system. So we have our reaction rates, these rate constants, and this these are called parameters in these mechanistic mathematical models, these polynomial ODEs. And we take their difference and then we multiply by the product of the species in the reactants. So let me give you a concrete example. So if we have 4 reactions and we have 3 species, X, Y, oh, no, I have X, we're gonna call this XW, XY, and XZ. Then we can look at the first reaction. And just by writing with this formula here, we can look at these are the reactants, and they form they form 2W, and then we can take the difference of the product and the reactant, and what, this is just a 1 in front, right? And that's 1. So then we have K1, and then the monomial XWYW, XWXY, and that is that term here. And then we can look at the next one where we see there's no W in the product. And so we see that it's a minus, so 0 minus 3, 0, so minus 3, the rate here, K2, and then our XW cubed. Okay, and so we can continue doing that. And then from this reaction, system of reactions, we have a polynomial dynamical system. So this is one type of mathematical model. And so in this talk, I first want to talk about single pathways. So this is the assumptions we're looking at, one single pathway. And then I'm going to talk about how to think about multiple different pathways. And then I'm going to talk about how to think about when you have these spatial patterns. In biology, they're often governed by, again, these molecular interactions. So we're really going to continue to build in the complexity through this talk. Okay. Great. Okay. And so I guess I just need to say it's a system of ODEs, and it can be helpful. This is a nonlinear system, but then there are linear. So you can think of this matrix is what we call the complex to complex rate matrix. So if we think of new species and on the left and the right, often people can look at this as a directed weighted graph. And so you can look at the negative, the weighted graph Laplacian is A kappa. So there's lots of nice things people work on in the linear algebra in the field of, there's a field in applied math that's been taken over by many real algebraic geometers, and that the field is called chemical reaction network theory. So once we have this mathematical model, a question is, how do we analyze it? And I would like to convince you that we really need imaginative mathematical approaches to study these biological systems. So this is basically the outline for the rest of the talk. So the first problem I'm going to talk about is the MEC-ERK pathway. And the geometry that's encoded there is looking at injectivity of a map, which is called the model prediction map, going from our parameter space to then our data space, So that is if we have perfect data. And then when we want to think about noise, we're really looking at the maximum likelihood estimate and looking at bounded confidence regions. I'm not going to talk about noise too much in this talk. So for all of these examples where things become more complicated, I'm happy to show you the references where this is discussed. Okay, so identifiability is looking at injectivity of a map. This is structural identifiability, which is a precursor for actually performing parameter estimation. So trying to recover, right, in these models, trying to recover the rate constants, these parameters. Okay. And then the second thing I'm going to talk about is data integration. How do we start putting different types of data, biological data together? And the geometry of that is looking at tensors. These are multi-indexed. So right there, higher generalizations of matrices. Many of you are probably very familiar. So we can have an adjacency tensor that explains what's called a multi, so we have tensors, but then when we don't have all of the data, then what we have is what's called a multi-layer network. And that's where all of the kind of layers of our data may not share exactly all the same nodes. So then the next problem that we look at is with all the spatial patterns. And many of these are looking at different types of tissue. And this, again, many of the examples that I present are related to cancer. And the mathematics is that we're looking at studying homology, and these are these persistence modules. And so for those of you who like geometry, these are finitely generated graded modules over a polynomial ring. And with one parameter persistence, this is in one variable, and in multi-parameter persistence, where we have more than one parameter, this is in multivariates. And so the question that as a mathematician we might want to ask is, can biology lead to new theorems? And that was posed by Baron Sternfeld some years ago. And I think it not only leads to new theorems, but it also, biology, new theorems can lead to new biological insights. So I really do think there is this nice cycle. Okay, great. So I mentioned, oh, I'm supposed to take that slide out. Okay, so here's the kind of first example, which is the kinetics of this MEK ERK signaling pathway. So I want to highlight, so Emily Dufresne, who's an invariant theorist at York, she specializes in separating variants, and Lewis Marsh, who's part of our Center for Topological Data Analysis, carried out most of the work in these projects. But it really started through a visit to a fruit fly lab in Princeton. And so Stav Schwarzman and Martin Wurr have labs at Princeton and fruit fly, and modifying some of these some of the mutations we look at related to MEK. And this is a pathway that just functions in a very large percentage of cancers, 65% or so of cancer, and many pharmaceutical companies and are very interested in this particular pathway. So we have a very simple model. There are hundreds of models of the MEK ERK biology. We have these different species. So we have a substrate ERK that is not modified, that is singly modified or doubly modified, and it's modified through an enzymatic reaction by what this enzyme called MEK. So there's a system of reactions that we can write down, and then we can reduce from the reactions to a polynomial dynamical system. And I haven't included all of the details, but we can reduce the model exactly as well as approximately. And so this is pulling out scaling symmetries, pulling out You can use differential algebra to reduce your model. And in our setting, we used what the quasi-steady state variety to reduce our model. And we did this by Gucka and Walcher's method, and we get a linear ERK model. So now we have a system of three reactions, three ODEs, and the biologists are very interested in this pi parameter. I didn't choose the names, the experimental groups did. But in particular, they care about the value of pi in this system of equations. The experiments that they performed was taking, it's a very simple, well, simple to explain. They have their enzyme, which is activated MEK, and they have a substrate, ERK, they add energy, and then they look at the unmodified, singly modified, and doubly modified species over time. They quantify that. So we have 3 species. This is our sample space, 3 species and 7 time points. And for wild type ERK, they do this 11 times. So we have different numbers of replicates. So our data space is our 3 species, which is the unmodified, singly modified, or doubly modified ERK. Our 7 time points are our replicates. And then they did this also for different mutations that are involved in developmental embryonic development dysfunction and defects, as well as in cancer. And then interestingly, they have the S222 or the SDD mutant, which is what they often will use instead of wild type as an activation. So they actually did activate it through the normal way, as well as with what is often used. And so we have data of ERK being activated by different mutants of MEK. Okay, so that is the mathematical model. And then our goal is to estimate the parameters. And so the idea, and when I say estimate the parameters, these are the 3 parameters that we want to estimate from this experimental data. Now this sounds very easy, but so we have 2 papers. We have a biology paper and then a mathematical paper, which kind of sets a little bit more of the mathematics. And the idea is that we have a point in the image and we want to know where it came from, right? So we have each model and experimental setup induces what we call a model prediction map. This is phi at the 7 time points, going from our parameter space to our sample space. And we let this data point, so this is our experimental data, said not be in the space of observable model solutions. So parameter estimation corresponds to computing the inverse of this map. And structural identifiability is determining whether our model prediction map is injective. So, so we can formulate the identifiability problem, ask questions about fibers of the map from parameters to, with these initial conditions, to the system of ODEs, so these measurable output data, which is some function of our corresponding solution. And it's very difficult to know how to do this for specific time points. So we can instead look at what's called an abstract model prediction map from these ODEs solutions. So this is the 3D space of real analytic solutions in our 1 time variable. And we can write out this equivalence relation, which is that we can say that 2 parameters are equivalent from our abstract model prediction map implies that 2 models are equivalent with our model prediction map. Now, by using the theory of differential algebra and differential ideals developed by Ritt and Kolchin in the 50s, 60s, 70s, we can translate asking injectivity about this map about this abstract model prediction map into looking at a different coefficient map. And this testing injectivity of this map is theoretically computable, and therefore we can then say something about whether we can recover the parameters from our model. And so I would like to highlight, right, so I'm going to give the definition of structural identifiability on the next slide. This was modified from Seth Sullivan's book. We developed this geometry of sloppiness led by Emily Dufresne. And then for talking about this particular setup with our ODE system, it's in this current preprint. Okay. So the definition of structural identifiability is that we can say a model is structurally identifiable, generically globally identifiable, if this is this map is 1-to-1, and then if it's finite to 1, then it's generically locally identifiable, and generically unidentifiable is when this is infinite to 1. So I guess the key is that we can have different notions. There's many additional notions of structural identifiability. But what we prove in our paper is that this one here, looking at the MEC ERK system, is that the linear ERK model with the experimental data that we're given, and that's really key, right, is that with the data that we're given, we can prove it's generically structurally globally identifiable and is practically identifiable. So with perfect data, we can recover these parameter values, and then with imperfect data up to some significance level, we can say that the pre-image of this is bounded. And so the MLE can be obtained with some bounded confidence interval at some arbitrary significance. One of the open questions we have is, how do we test structural identifiability with specific time points rather than there's a theorem where we have to have a sufficient number of time points from Eduardo Sontag, but actually, instead of saying generic time points, specific ones. And so that's an open question. Okay. So then we can use Bayesian parameter inference once we know that our model is, excuse me, once we know that our model is both structurally and practically identifiable, we can infer the parameters, and we can do that for each of the different mutants. So let me, so I guess I'm assuming everybody knows Bayesian inference, that you want to infer the parameters in light of the data. And so a statistical setting is that we use the likelihood, and we have a likelihood function because this is a deterministic ODE, and maximizing that likelihood gives us a value of the parameters, and that maximizes the probability of observing our data. And so we assume that we have, and this is with collaboration with Stas and some experimental values in the literature, that we have some range. And then we can infer these three parameters. And so I guess what I should highlight, right, is that the black one here that's the wild type, and the orange here is the mutation that's involved in cancer. But we also see, because they have a fruit fly wing lab, that we have dysfunctions in the fruit fly wing. But we also see that this SSDD mutant, while it's not very interesting in their parameter pi, it is very different in the other two parameters. And so we wanted to ask how to compare the kinetics of these different MAC mutants, and there's many different ways to compare these posterior distributions. One way that we did this, and this is our first application, it took me a little while to get to, but the first application is computing persistent homology of the parameter posteriors. And so we have a posterior distribution here. We have in the form of samples, and we can estimate using a KDE of this posterior density, and we approximate the persistent homology of super level sets of the kinetic, these parameter posterior densities, and we use that with simplicial complexes, and then we compare them with the output of the barcode with the bottleneck distance. And in particular, we implement this work by Omar Borovsky, Shayan Mukherjee, and Jonathan Taylor for this topological consistency via KDE. And oh no, I had a movie. Okay, I should have checked this, I guess, before. But basically what I was showing you is that we have this super level set filtration, and it outputs a barcode. You already know what a barcode is because I explained it in the first slide, but it was just showing you kind of a density function and giving you what the corresponding barcode would be. And then we can compute the bottleneck distance between barcodes. Again, Kate's going to go through that in more detail, but what we find is this SSTD, what's normally the constitutive active mutant, is a very large difference in bottleneck distance than the other mutants. Okay, so that is the first example where we're looking at one single pathway. Now, the next couple examples is integrating multiple pathways. And so once you start having, instead of so we looked at the RAS signaling, which is the MEK ERK here, and we start looking at other pathways. So for example, first glance, it looks like all of these are disjoint networks, but actually they're not. So if we kind of zoom in, you can see that MEK and ERK appear in these two different signaling pathways. And so one question we can ask is what properties hold when we join them. So these vertices would be actually, we would join them, but maybe we want to split up the reaction. So when does identifiability hold, or another property that we're interested in chemical reaction networks? And so we have a series of results on looking at, starting to integrate these different multiple pathways. So this is sometimes called crosstalk. Now, motivated by exactly this ERK pathway, Merrimack Pharmaceuticals funded this study where they had 3 people spend an entire year generating the concentration of different species in the ERK system, as well as the AKT, which is involved in insulin, but also dysfunctions in cancer. So they had 36 breast cancer cell lines. And remember that in at least in breast cancer, they know there's certain mutations associated with certain breast cancers. So there's some of these have mutations in them, but we don't know a priori. So 36 breast cancer cell lines and 14 ligands, and these are signaling molecules, and then different doses of the ligand. And we want to understand how the 5 variables, the cell lines, the ligands, the doses, the time points, and these species, so we have 5 variables, are related. So one way to extract interpretable structure is to integrate it into, taking this multi-index data and integrate it into a tensor. So in their previous work, the Merrimack Pharmaceuticals, so Birgit Schobel and her colleagues, they flattened this to be cell lines and everything else. So this was a 36 by 224. Actually, they kind of only took a slice of this, so they didn't include either a low or high dose. But you can use linear algebra tools, and then you lose that structure. So we want to incorporate the multi-index structure, but also cluster it. And so we first did some initial clustering with the 36 cell lines and 14 ligands. And you can kind of, if you squint, see that there's some structure here. It looks like everything's in one group here, but then there's a couple different groupings. So this was some initial clustering with 3 different clusters. And then what we would like to see is that there's some rectangular, so there's some constraints that biologically, that we can encode, they mean something biologically. And so we take we have this partition tensor. And so we start with, there's a 1 if the cell ligand is in cluster C. So C is 1, 2, or 3, or 0 otherwise. And then we want to find a new clusters that, so we're maximizing the inner product between our partition tensor and X, which is what we want our new clusters to be in the 36 by 14 ligand by 3 different clusters. And we used Cplex integer programming optimization to do this, where every cell ligand is in the unique cluster, but then this is also rectangular. Okay, so we perform this, and we get out these interpretable groupings, which then we can go and construct. We can interpret it. So the first thing that's pretty cool is that when we looked at certain certain cell lines, we know there's certain mutations, and some of the clusters all had the same mutation, which we didn't know a priori. Or some of the clusters, the ones that were had these types of receptors that bind to these ligands, there's something special about them. So without knowing any of that information, our clustering kind of pulled some of that out, and it improves the current classification, which they have of breast cancer cell lines. So I think that's quite exciting. And this was work with Anna Siegel, who's an expert in geometry of tensors. Now, building on that, during the pandemic, there was work where we had patients in the hospital here in Oxford, and they were hospitalized COVID patients, and they could be classified by the World Health Organization classifications with different, it's pretty coarse, but the amount of oxygen that they needed. So with COVID, mild, severe, critical. Then they had community COVID-19, which are the healthcare workers that were getting COVID, then healthy volunteers, as well as initially this was studied for sepsis and flu. So we had other information, and they basically collected everything out of the blood. And this is where I, in 2020, was learning about genomics and omics and multi-omics and a very high dimensional data world. So we used tensors, we used topological data analysis, in particular an algorithm called Mapper that I'm not talking about today, and machine learning to be able to cluster and classify, excuse me, some of the patients by some biomarkers. And so this was a paper that was published at the beginning of the year in Cell. And we've built on this work with a PhD student, Piotr Sliwa, and this is work in prep that he's presenting at Complex Networks. And the idea here is that you can have the proteomics or the cell type abundances and gene expression, single cell gene expression, and create a network where each node, so you kind of have different layers, which is not a necessarily the layers you're thinking of in machine learning, but these are different layers where each layer represents one of the modalities that we have. And each point here is a sample, so it's a patient sample. And then we can construct, normally what one does is just analyze only one type of data. But now we have data for patients, but we don't have all of the data. So some data we don't have measurements of. We know that that person is a sample, but we don't have a measurement, for example, of some cell abundance. So there's a multi-layer network rather than a multiplex network where you have a complete data set. You have for every entry, you have all of the modalities, which is what we have for COVID patients. But once we start to extend to all the patients, we don't have that. So we can then use a multi-layer network approach and find clusters, communities, and that seems to improve the classification as well as finding different biomarkers. So that's exciting work that we have. Okay, so now the rest of my talk, I want to highlight other areas of TDA. So far what I've introduced some computational topology, then I introduced one pathway, a single pathway, and how we can analyze it in this parameter identification problem. I tried to include a little bit of geometry in there since some of you may be interested in this, and then multiple pathways and how we can start looking at some geometry of tensors, right? So in the first tensor example, we talked about tensor clustering. In this tensor example, we were computing components. So we were computing decomposition of our tensor. And then here, when we have more noise, what we've been doing is some community detection to find, again, some clusters. Now, the rest of the talk is just a couple slides for each example. And it's looking at different spatial temporal models and data. And so there are many different types of models. And so in the examples, I'll talk about with many collaborators, and I want to highlight the early career researchers because this is a school, and I want to highlight that even in your final year of undergrad, you can do really cool and exciting research. We have 4 or 5 undergraduates that are doing research in TDA that I know about, some in Oxford and elsewhere in the UK this summer. So the first project I talk about is looking at protein structure, and this is knotted proteins, and that's with Agnese and Kate Benjamin. And the second project I talk about is looking at images of bone fibrosis, and this is with Jill Grindstaff and Bernadette Stolz. Then I will talk a little bit about some work that Bernadette did looking at the shape of abnormal tumor vascular networks. Then I'm going to extend to some of this vascular network, so you can think about this network. It's a graph, and sometimes there's graph classification problems. And so we've been starting to use other notions from one parameter persistence to other types of persistence, and this is Jacob and Ambrose. And finally, going into immune cell locations, and that's work by many people, but Oliver Vipond and Joshua Bull. So I have about 2 minutes per example, just to give you a feel of the many different ways that we can use persistent homology to study spatial temporal models and data. Okay, so so far I've mentioned there are many types of models, right? So these chemical reaction network polynomial models, these are ODE models. Then when we think about space, there's PDE models. If you speak with a biologist, they think of a mouse or some organism model. But I'm going to start with some of us here are topologists in particular. You might like low dimensional topology. So one of my supervisors was Dorothy Buck. She was at Imperial, of course, part of the London network, and her expertise was DNA topology. And so I was really inspired by her. And so we looked at, instead of closed knots, right, which is how we think of a knot, there are open knots, and these occur in proteins. And so there's a small fraction of backbone knotted proteins. And these knots can be deep or shallow, and that's defined by kind of the relative length of tails versus the core. And so if we look at trefoils, trefoil knotted proteins, one interesting thing is that the stability of the protein increases when they're knotted. So one question in general, the motivation is, what's the structure and function of knots in proteins? So we look at these open-ended trefoil knots. Can we distinguish almost exact structure? So if you look at this backbone, the carbons, right? And we can look at this, the protein, this is from the PDB database. They look at the sequences, they're called sequence homology. The sequences are almost exactly the same, but some are knotted and some are not. And so we have developed this pipeline where we take in this piecewise linear open curve, which you can think of as a point cloud. So we have these set of points, and then we can grow epsilon or t, radius t balls around each of them, and we can compute the homology, so the persistent homology as this t increases. And we use Vittorius Rips here. So we have a backbone protein. We compute the persistence diagram or barcode. We can compute topological statistics from it, and we can look at what are called generators, so that if we take one point in our persistence diagram, we can say what vertices were involved in this. And this is not a unique thing that we can do, but there's a software called Irene, which allows you to look kind of more locally at what the TDA gives you. So this is helpful for interpretation. And what we find is that we can take the average landscape, persistence landscape of knotted and unknotted proteins, and we see there's this little blip here. We can look into it, and actually what's different, the localization, is this crossing change from being knotted to unknotted is just that crossing change. And so we can look at these different proteins and very clearly see, but based on the landscape distance, that the knotted and the unknotted proteins have much similar persistent homology. So it's one example, and that's again Kate and Inez's work. Another example that I'm really excited about is work that Dan Royston and Hosek Raju, they were looking at bone marrow samples, and they were with machine learning creating a continuous classification score. So each pixel here is given a classification, given a value for how fibrotic that particular pixel is, and they use machine learning. So this is bone marrow samples. These are from patients with myeloproliferative neoplasms, and that can develop into acute myeloid leukemia. And they have several different diagnostic subtypes. So they look at this and they say, oh, that's pre-PMF, and they look at this one and they say that's ET. And so they wanted a continuous score classification with these small regions, and the ML output from this continuous classification is from this image. They can quantify this into a feature vector for subtype classification. And our question was, could TDA help? So we used, instead of, we used super level set. So that was what I wanted to show you the video of, and it didn't work. The super level set filtration. So you're including in your filtration and building your simplicial complex with these level sets. And what we can see is with pre-PMF versus ET, we can find some of these components, and then we see where they merge. And so we have a most persistent loop here, where we can see that here. So we can look at the loops and as well as the connected components. And these, the birth and death and all of these different possible topological features are put into random forest, where we use the original original vectors and the topological feature vectors. And we train this data, we do random forest classification. And what we find is that, that in general, the topological descriptors improve their classification. And so this is available on medRxiv from a couple months ago. Okay, so then from this, right, as I mentioned, that this can be the fibrosis score. So these different subtypes, it really matters because it can be a precursor for cancer. And so, oh, I'm almost out of time. So I'm going to say 2 minutes. These cancer data sets, we've looked at what you can look at as blood vessels. And these blood vessel networks are abnormal, as well as immune cell locations. And one of the questions is, how do you find a meaningful filtration from your blood vessels to your TDA output? And we took inspiration from many existing work. Some of this is here in the UK, right? This is Jacek Brodsky's work, or neurons, and looking at from Kanara and Catherine Hess. And we created this filtration from the center of the tumor out, which we can quantify and interpret. For example, the number of loops, which you have in near tumor vascular networks. And we applied this topological method on different mouse models, and we show that if they applied a treatment to reduce the number of vessels, blood vessels, that you have a reduced number of loops. Just like if you apply a treatment, you kill some of the vasculature and you have an increase in the avascular, so these voids. There are many different mathematical models. So I want to just, in the last couple of minutes, talk about, so our chemical reaction networks, now you can think about them with space and add stochasticity, and these are agent-based models, and they generate data. And so we can, instead of using homology and just get the number of loops, we can use persistent homology and get the loops and the location, and then be able to classify into kind of different groups. But what we really want is the number of loops, the location, and their size. And so we started doing this inference problem on using this specific model to be able to recover the parameters. And we used extended persistence. Now there's some other work, so let me just tell you a little bit about extended persistence, and then I will wrap up. So extended persistence, as I said, it's a way to give you the size, the loops, the location, and it's an invariant of these piecewise linear function. And so we can get an extended persistence diagram, which then can be vectorized into a vector representing the topology of this filtered graph. And what I want to just mention is that this has been used in machine learning for graph classification. I realize I'm running out of time, but I would like to make a plug that extended persistent homology, which was done by two early career researchers in Oxford, so Ambrose and Leone and Jacob Leone, were able to include extended persistent homology for this graph classification. So this is where it's used as an optimization layer in a neural network architecture. So it's not only just for inference, which is how we use extended persistence, then they used it in graph classification. And then the final application, which I won't have time to talk about, is how we can use multi-parameter persistence. So I'm just gonna maybe mention there's this great review article by Magnuson and Mike Lesnick to look at multi-parameter persistence. And we were able to use multi-parameter persistence to not only look at structure and shape, but also start to integrate different types of data. And so I'm really excited about the prospect of being able to, with additional notions beyond one parameter persistence and other areas of computational topology, to be able to go from a biological hypothesis to various notions of models, to their analysis, and really exploiting the data analysis machine learning pipeline to inform them. Okay, and on that note, thank you very much for your time.